Hello everyone, this is Dr. Cary Harding. It's coming to you from Portland, Oregon. I'm a professor of genetics at Oregon Health and Science University, and I'd like to wish you a very happy International PKU Day. I was asked to make some comments about the status of research in the PKU world, and particularly on the status of new developments in novel treatments for PKU. I have a second task today, and that's to begin preparing a meal for my mother-in-law's 80th birthday, which is tomorrow. And she had a very specific request for her birthday meal, and that is for smoked brisket. Now, I know that most patients with PKU cannot enjoy smoked brisket, but as long as I had the task to do to prepare this, I thought I'd have you come along and help me with this anyway before we start talking about PKU. So it might seem kind of cruel for you to have to participate in this endeavor, since I know many people with PKU can't participate in this, but I have to fill the smoker up about every half hour or so to keep the smoke going. But it got me thinking while I was having to multitask these things that one of the goals, obviously, for some of what we're trying to do in the lab is that perhaps one day an individual with PKU would be able to partake in that barbecue, although that's not really the sole motivation for what I do. The goal, obviously, is to allow someone to liberalize their dietary protein because I think that's healthier for them, not just because I think it's fun, but I do think ultimately it's healthier and better for their brain to be on a more liberal dietary protein intake and still keep their phenylalanine levels down. So this double task today got me thinking about the specific focus for my lecture today. So that brisket needs to smoke for hours. So while that's going on, let's talk science. The formal title of my presentation today is Novel Therapies for PKU. But the real question I have on my mind today is, when do we get to barbecue for PKU? These are the topics I would like to cover in this presentation. There are certainly novel treatments and PKU researchers that I simply do not have time to discuss today. To those who I have left out, I apologize in advance. The first new and revolutionary treatment to discuss is Pegvaliase or PegPal. Although novel, it's already been approved for use in the US and in the European Union. This is the first treatment that has the potential to both lower blood Phe and at the same time allow increased dietary Phe intake. It is a bacterial protein coated with polyethylene glycol or PEG to help protect it, and it's injected daily under the skin. It is an enzyme that circulates in blood and metabolizes Phe. Because it's a foreign protein, everybody makes antibodies against it. Those antibodies can cause skin rashes or joint pain in many people, but also more severe reactions in a few individuals. We've learned how to prevent or manage these reactions by starting with low doses of Pegvaliase given very infrequently and then increasing the amount and frequency of dosing very slowly over time, and then we also treat with antihistamines prophylactically. Most people with PKU ultimately are able to get to past the hypersensitivity reactions and achieve a dose that lowers their blood phenylalanine. We've studied Pegvaliase in numerous clinical trials over 10 years prior to its approval for commercial use in the United States, which happened in May of 2018. To date, there are now over 500 adults with PKU in the US who are being treated successfully with Pegvaliase. Approval in the EU came about a year later, and treatment is being slowly rolled out in Europe. Professor Anja Muntau in Hamburg, Germany, has been leading the way there and has treated about 11 adults with PKU so far. There are two new oral treatment approaches that have recently entered clinical trial. Synlogic has developed a probiotic bacteria that produces an enzyme which is similar to Pegvaliase. In this case, the patient would take capsules containing the probiotic orally with each meal. And these bacteria will pre-digest some of the phenylalanine in the food before it can be absorbed by the gut. Nestle has licensed similar technology from a company called Codexis. They've engineered a PAL enzyme that is stable by itself in the gut if taken orally. This would have a similar treatment approach to the probiotic. You would take some of the enzyme orally with meals to pre-digest the feed in the food. Both of these treatments are currently in clinical trials in adults with PKU. Neither approach is likely to be a cure, but they may help significantly increase one's dietary Phe tolerance. So there's been a lot of hype for many years about gene therapy. And we're finally to the point of actual clinical gene therapy trials in PKU, so this is a very exciting time. So what is gene therapy? This is simply using genetic material, which is most commonly DNA, or deoxyribonucleic acid, as a drug to treat a disease. So how does this work? So on the left here, I've drawn a depiction of a typical liver cell that contains two copies of a normal PAH gene, PAH standing for phenylalanine hydroxylase. We inherit two copies of every gene, one from mom and one from dad, and the PAH gene is the code that provides the instruction for the liver cell to produce the enzyme phenylalanine hydroxylase, or PAH. PAH converts the amino acid phenylalanine, or Phe, which we obtain from protein in our diet, into another amino acid, tyrosine. On the right is a liver cell from an individual with PKU. In this case, both PAH genes carry changes which we call mutations. These mutations alter the code and either prevent the production of PAH enzyme. In this diagram, Phe is not able to convert to tyrosine. Phe accumulates in the body, and at high levels, even though it's a normal amino acid, it is specifically toxic to the brain. The most straightforward form of gene therapy is called gene addition. In this approach, we don't actually alter the patient's PAH gene. We just add in a normal copy of the PAH gene to the liver cell. There are a variety of ways to do this, but the treatment that has been done most successfully takes advantage of nature's best gene therapists, which are viruses. The natural function of a virus is to infect a cell, and they carry their own DNA into the host cell. The virus we work with is called adeno-associated virus, and we'll abbreviate it AAV8. We have engineered this virus to carry a normal PAH gene along with some bits that are necessary to tell the liver to turn me on and make PAH. We can make the virus, which are small particles in this picture, inject these into animals or humans with PKU, and they will infect the liver to make PAH. We have a PKU mouse that has been our model system and test bed for all of our novel treatments. So after a single IV injection of our engineered virus, the liver begins to produce PAH, and the blood Phe levels in the mice immediately plummet into the normal range. High blood Phe inhibits the production of melanin, the pigment that gives us and mice hair and skin color. So PKU mice are tan-colored rather than the expected color of black. Once their blood Phe decreases, their coat color turns to black. So after a gene therapy experiment, we don't even really need to do the blood Phe measurements. We can tell who was cured by the color of the fur. We did this almost 15 years ago. So why hasn't there been a human clinical trial yet? There are lots of reasons, but the biggest has been waiting to accumulate data on safety in humans with other diseases. That has now happened. And finally, there are two different companies who now have active AAV gene therapy trials open for adults with PKU. Homology Medicines, which is a company in Boston, announced in December 2019 that they have treated 4 PKU adults with a particular flavor of AAV they call AAV-HSC. And this is a small trial that started with very small AAV doses and then escalated to higher doses. In at least one of the subjects that got a slightly higher dose in the second dose cohort, the blood feed dropped by about 50% 4 weeks after treatment. These are very encouraging initial results. There's not been a subsequent press release. BioMarin has their own flavor of AAV called AAV5, and they were well on their way to starting a clinical trial when the COVID-19 pandemic hit. So this has slowed things down a bit, but we're hoping that recruitment and treatment plans will resume quite soon. There are a few other companies with plans to start gene therapy trials in PKU with different flavors of virus and different approaches, but they haven't specifically announced their trial details, so I am not listing them here. It's impossible at this point to predict whose vector or which approach will be best. There is a fundamental problem with gene addition. Liver has the unique capacity of being able to rapidly heal itself after an injury. Normally this is very beneficial for us, but it might not be so much if it's after you've had gene therapy. The liver cells in adults gradually turn over and are replaced, perhaps at a faster rate if you are fond of a good single malt scotch like I am. With this gene addition approach, the piece of viral DNA in the liver cell does not associate in any way with the genetic material of the cell itself. If something causes the liver cell to divide, the cell's genetic material is copied, but the gene therapy vector DNA is lost. In the mice, if we do something to damage the liver after gene therapy, the remaining liver begins to be repaired and the gene therapy is immediately lost. We can't give another virus injection because the mouse or human patient will have developed immunity against the virus and will destroy it the second time around. We do not yet know how long AAV gene therapy in adults will last. Furthermore, this approach won't work in young children because in the growing liver, the liver cells are dividing very rapidly and they quickly kick the viral DNA out. We know this from many animal experiments, including in PKU mice, in which we lose the gene therapy within a day or so after we inject it into newborn mice. So how can we design a more permanent gene therapy? And this is the current focus of our lab work. What if we could find a way to go in and physically fix one or both of the mutations on the patient's PAH genes and restore their own PAH production? This is called gene editing. There are a few different ways to try to achieve the surgical correction of a mutation, if you will, but one of the hottest technologies in the field now is gene editing using CRISPR-Cas9. You might have read about this or heard about it, or your kid might have already tried it at preschool. Everybody's doing this. Here's what we've done with it. We've taken the same AAV8 virus and swapped out the PAH gene and put in the gene for Cas9. Cas9 is an enzyme that cuts DNA, but it's very discriminatory. It seeks only very specific DNA sequences and cuts only at that specific site, but no other. The beauty is that we can engineer the system to cut in a very specific site close to the mutation in the PAH gene and cut only there. Cas will frequently cut both chromosomes, but I'm just showing it cutting one. Now Cas9 just makes a cut and leaves a hole. Somehow, a lot of people, even a lot of scientists, have the misunderstanding that CRISPR-Cas9 editing cures genetic disease. But really, it causes genetic disease. It just makes a cut in the DNA. It doesn't fix anything. The cell itself has mechanisms that can fix the hole. We've learned that the liver, unfortunately, prefers to just take the two ends of the hole and stick them together and patch things up. That doesn't cure anything. In fact, it makes things worse because now you've just basically wiped out a big chunk of the PAH gene. How do we fix this? We infect the cell with a second AAV, but this one carries with it a fragment of the normal PAH gene that matches the area that was cut. We add this fragment, and our hope is that in the course of repairing the hole, the cell will incorporate this piece of the PAH gene. If this is done correctly, the patient's chromosome now has an intact, full-length PAH gene without any mutation that encodes the correct sequence for PAH protein. So we learned early on that unlike gene addition, this only worked if we applied it to baby mice. If we waited until they were adults, we only see deleted PAH genes and no corrected genes. So we injected infant PKU mice with mixtures of both AAV vectors, the one that makes Cas9 and one that has the repair template. And it turns out we had to do an additional pharmacologic trick. We had to treat them again as teenagers or adolescents, and then we finally waited until they were adults to analyze them. So this is what we did. Treated them as pups, weaned them, treated them again, and then as adults we examined them. And the great news was that some of the mice as adults had turned black, looking like their unaffected litter mates. But we learned that we had not actually completely cured the mice. The blood Phe had dropped tremendously to over 60%, or we had a more than a 60% decline from where they would have been if they were not treated. But their blood Phe levels did not completely drop down to normal, which would be down to the green, which is the level of where a wild type mouse or a normal mouse would be at. We found out that we had only corrected about 7% of the liver cells back to having a normal PAH gene. We know from other work that we need to get over 10% in order to completely correct the blood Phe down to normal. But this is at least a very promising start, and we are now working to find ways to increase the number of gene-corrected cells in the liver from this treatment. There is a drawback to CRISPR technology as we have used it. The reagents must be designed specifically for every mutation you target. So there are over 1,000 known PAH mutations in humans, and this potentially means that you'd have to design 1,000 different drugs for every different mutation, so 1,000 different drugs. This really is very much impossible, but we have another idea that we're currently working on. This is a combination of addition and editing, where we insert a full-length PAH gene somewhere into the human genome so that it will be permanently present even if the cell divides. In this first attempt, we're using a different virus called a lentivirus, and it integrates itself into the host cell DNA, usually not the chromosome that carries the PAH gene. And it starts to produce PAH enzyme. The key again will be to achieve integration of viral genome into at least 10% of the liver cells for this to work. We are nearing the completion of this experiment, which we are doing in collaboration with the laboratories of Dr. Marcus Grampe at OHSU and Luigi Naldini in Italy. This is our overarching goal: to develop a safe, effective gene therapy method that will permanently produce PAH enzyme in a majority of liver cells, leading to permanent reduction of blood phenylalanine, even if the individual with PKU is consuming normal amounts of dietary protein. There are many people in my lab and in several labs around the world with whom I have collaborated over the years who deserve immense thanks for assistance with this work. I have been very fortunate to have received very generous funding from the National Institutes of Health in the U.S. as well as the National PKU Alliance. But the NPKUA is also currently funding other investigators who are working on various aspects of PKU, both in terms of novel treatment, but also trying to understand further aspects of PKU and disease, and these are the current other investigators that the National PKU Alliance is funding. I was also asked to comment about the status of home portable Phe monitor development. Currently, there are 3 different types of devices being developed by 3 different companies, and all of these look quite promising. All are about to begin clinical trials in PKU patients. Fortuitously, NPKUA has just hosted a webinar in which all 3 companies participated and described their progress. And this webinar is available for viewing on YouTube. Finally, I want to mention the establishment of a new research consortium, the Phe-Free Consortium in the U.S. Phe-Free stands for Phenylalanine Families and Researchers Evaluating Evidence. This consortium grew out of the Pegvaliase clinical trials with the goal of continuing to study long-term outcomes in individuals of all ages with PKU, but also other genetic disorders that cause elevated blood Phe, such as the diseases that affect biopterin metabolism. The consortium has been in existence for less than a year, so we're still establishing our research priorities and protocols. And we are funded by the U.S. National Institutes of Health and have a very close partnership with NPKUA. Once we're on our feet, we hope to expand to other centers, including to international partners, and our ultimate goal is to form a large clinical trial network that will facilitate research for novel treatments in the disorders affecting phenylalanine metabolism. It is certainly our hope that one of these novel treatments, or something maybe that we haven't even thought of yet, will one day significantly improve the lives of individuals and families who are affected by PKU. But very much like waiting for the brisket to slowly smoke on the barbecue, these breakthrough treatments will only come with time and with patience. I thank you very much for watching, and thank you for supporting the activities of International PKU Day.